as a pack. They have such an extraordinary degree of intuition that their understanding of one another's intentions often seems beyond normal explanation.
Come to Brightown, this is my town.
Living my life, living my life. Living my life, living my life. You wouldn't last. Living my life, living my life. Living my life, living my life. You wouldn't last. Till it blows, legs MC, that heart of gold, carved in stone. Running your gums, you better park it slow. Just some things that I can't condone. Been in the game for 8 years, making rap tunes. Ever since you were flapping in chat rooms. Living back on Queensway, man's are trapped in cliché. Kick back, rely on the DJ. Unemployed, getting fucked on a weekday. Been there, got the t-shirt on eBay. Got that shit out the way in my teenage. One day I'll be a brilliant man. Tech microphone like Millie and Dan. Crystal clear like a digital scan. Then disappear like a visible man.
One day, when the world's in our palms and we can escape to another place. Get high on a life of rhyme, living in a state of mind of a deep place. Cause we are the chosen fam. And any head who don't lie, we don't give a damn. Just living our days in a dreamland. And we're never gonna stray from the main plan.
Living my life, living my life. Living my life, living my life. You wouldn't last. Living my life, living my life. Living my life, living my life. You wouldn't last.
Mr. White with the conscious words. I follow my heart, not follow the herd. Speak slow, slurring my verbs. Living on coffee and potent herbs. You can't fuck, cause this is the proof. Legs are white like a bomb in your roof. Don't look now, I'll spit on your mask. Step on your groove, blast them apart. Look at me now, read my fist. Born in this fix, I ain't ripping no shit. One of these days I'll make myself rich and get a grip on this nervous itch. Cause I'm getting bored of spitting to a brick wall. Ready for the fat shows and the grand halls. Waiting in vain for the applause. Out in the way by the seashore.
We stand tall among these posers. When the smoke clears and the show is over, I get cold like Nova Scotia. Ain't taking shit from no promoter. Bag of chips all over my shoulders. Trigger the bombs, now flows exploding. I'll be rapping till my throat is closing. You drop your guard, now your nose is broken. Living my life, living my life. Living my life, living my life. You wouldn't last. Living my life, living my life. Living my life, living my life. You wouldn't last.
Just exploring a few of the claims. The mainstream media narrative is that it naturally came from nowhere, as it's racist to suggest it came from China. The UN and China did a great job for humanity. The right-wing narrative, or you could say QAnon claim, is the Chinese, the deep state or communism created it in a lab to destroy America and take over the world. But it's distinct polybasic furin backbone rules out purposely creating a bioweapon from scratch, so there's no way that it was created in a lab. A number of ways you can prove this. Exosomes, which was Andrew Kaufman's claim. No viruses have ever been isolated. They don't meet Koch's postulates and are exosomes. Koch's postulates, he abandoned the first and the third part, and they were invented 8 years before viruses were even discovered. So for people to still be trying to throw Koch's postulates, something that was created 150 years ago and was used for maybe 30 years, it's one of the biggest schoolboy errors, and it's quite funny. Next claim was the Kawasaki claim by Shield Destroyer. He went from it's 5G to Kawasaki to it doesn't exist within a year. Next claim is empty hospitals, which was, I think, an internet creation. The hospital wards were empty all over the world. Most of the people dying were in care homes or at home. Plus, it's not a third world country where people are treated in the hallways. COVID-19 wards are sectioned off from the rest of the hospital and have security. People running around hospitals filming empty hallways created this narrative. I think Debbie Hicks was one of the main people behind this. CDC didn't have an isolated virus, so the virus is not real. Well, this is a totally misunderstood and cherry-picked claim by, I couldn't tell you who, but basically they're using a test that the CDC did as early as March and claiming that this was the main test around the PCR test. That particular document is describing what was used to determine the lowest amount of viral genetic material that the RT-PCR assay could detect, and the limit of detection of the RT-PCR assay was determined. CDC have used transcribed RNA as the positive control, which means they use synthetically produced genetic material to that carried by the virus, meaning they are using something that has been synthesized and isolated already to determine the limit that the PCR test could detect. So again, totally cherry-picked and misrepresented in so many ways. The next claim is by Peter Bjorger, Mike Yeadon, and mainly Andrew Kaufman. PCR tests are designed to detect exosomes, chromosome 8, or that the amount of cycles stipulated by the WHO means that the virus isn't real. Again, this is another misrepresentation. Using the amount of cycles has nothing to do with detecting the virus, and the more cycles actually you do, the less chance of you seeing the virus because of the fluorescence, which I haven't heard any people mentioning or talking about in their claims when they do mention it, or their understanding of it. The next claim is the 5G claim, which would have come from either Thomas Cowan or Mark Steele, and what they were saying is 5G was first switched on in China, it poisoned the air and people breathe, causing them to die, not COVID-19. Again, like the empty hospitals claim, 80% of the people were dying in care homes which don't have 5G, and many people are dying in rural areas, which a lot of these rural areas don't have electricity, let alone even 3G. So to claim that all these people are dying from 5G is another pseudo misunderstanding by Mark Steele and Thomas Cowan. And then the next claim was the old man collapsing in the street in China. Why are people not dropping dead all around us? Well, number one, there was never a timestamp on this video of the man collapsing. Number two, we don't even know if it was in China during the time of when it was meant to be filmed. Also, to say that video from the mainstream media is proof that the mainstream media's virus doesn't exist it's another schoolboy error. What about his autopsy claim? Can you trust the mainstream media? Or what about China's source? People are just sharing this around as if it's some legitimate proof as to it existing. So there's many claims going around, and as per usual, virus denialists and the people making these claims don't have any test or way that someone like myself or a professional scientist could come along and test this to see if it does prove to be real or not real or legitimate in any way. To just throw words around based on a YouTube video that you've watched or a few YouTube videos that you watch is baseless claim. The next claim was people are dying with, not from COVID-19. Whilst this is true in some cases and death numbers can be and have been manipulated, people are cherry picking data and presenting it in their own way. Is it real? Does it exist? Some people, without truly understanding, say it doesn't. To all those who claim SARS-CoV-2 or any virus does not exist, the virus sphere consists of 4 realms, 9 kingdoms, 16 phyla, 2 subphyla, 36 classes, 55 orders, 8 suborders, 168 families, 103 subfamilies, 1,421 genera, 168 subgenera, 6,519 species. They've all been verified in a number of ways, and we've seen them growing in cultures. They've also been replicated many times by many people, even amateurs. This is the first transmission electron micrograph of the SARS-CoV-2 virus particles isolated from a patient in Russia in January of 2020. Here's the coronavirus successfully growing in a lab. Many people have done this as well. It was decoded in January 2020, just weeks after the world started to learn of the coronavirus. Each of those individual letters is a genetic building block known as a nucleotide, and when built up they form an organism's genetic code which we can use to understand them. Each organism has a different code and varying amount of nucleotides. A human has about 3 billion, whereas a virus such as SARS-CoV-2 has about 30,000. Thermo Fisher Scientific, it's one of the hundreds of websites that sell different parts of the SARS-CoV-2 that's been isolated, and you can buy different parts of the proteins. There's another website, BEI Resources, which sells every single virus, fungi, bacteria, everything that has been isolated, viral cultured, recombinated, and many different processes it's had to it. You then have another website with the whole purified viral lysate of the SARS-CoV-2 for 320 to over £1,000, isolated from a patient straight up from China. We've also got the human ACE2 is for sale, 230 odd pounds to £800. Isolations around the world. I stopped saving them after about 10 of them because there's so many you can find. There's also studies and scientific experiments on these links of the size of each individual parts of the genome of the SARS-CoV-2. We've got the number of proteins, the length, the evolution rate, the mutation rate, replication timescales, entry into cells, eclipse period, host cells, and it's all backed up with scientific reports and experiments from years ago or this year.
The point is, before I even talk about it, by the way, so that means they don't even care about the information. If anyone anywhere is saying Wuhan didn't start well, then you're just a shill. That's called a lack of intelligence, right? You're choosing to believe in something because you want to, not because it's actually verified, right? That's what children do. Now, in this case, we're staring at evidence. Doesn't mean we know it started in Wuhan, doesn't mean we know it started in the United States or anywhere else. It means that at the end of the day, we see evidence showing that there are clear financial ties back to the US government, to the very people who started the narrative. That has to be considered. And if you think that's being a shill, well, then you go back to your echo chambers, because you're never going to find any truth if you're not willing to consider things that you don't want to look at. I mean, we all have people that we believe in. We all have people that we want to trust. At the end of the day, you still have to be open to things that may paint them in a bad color. If you're not, well, then you're never going to be open to truth.
SARS-CoV-2 is an enveloped virus which has one of the largest genomes at 30 kb. It has an illicit lifestyle in 3 stages. Firstly, with the attachment and entry, replication and synthesis, and viral assembly and release. What happens is the spiked proteins attach to the ACE2 receptor through endoptosis. The furin of the TMPRSS2 is cleaved, and then the fusion of the virus of the host cells happen. The ACE2 is situated many parts of the human body, and once the virus is into the cell, the RNA is released. The viral genome is the positive strand RNA. The RNA is subdivided into 2 parts, the ORF1A and the ORF1B, primary translation being the first, and the structural proteins, subgenomic RNAs, being the latter. The S stands for spike proteins, the E is envelope, the M is membrane, and the N is nucleoplastic. The first section of PP1As make one long protein in strands of 10, but they need to be cut up before they can do their job. The cutting up happens with the 2 important proteinase. The PL proteinase are the ones shown in purple, and the 3CL protease in blue. So once the section is translated, the PL and the 3CL cut themselves off and start cutting off the proteins within the section. Once the RDRP is made, then they can start creating the RNAs that is needed for making the new proteins. Another classic that's going around this year is Koch's postulates. 2 of the most frequent claims made by virus denialists have been that the PCR test doesn't test for a virus, but the human genetic material, that was David Icke, and the classic Koch's postulates have not been met for the virus, that was Kaufman. As for Chromosome 8, on the 24th of August 2020 on Kaufman's YouTube channel, he claimed the PCR test used widely for testing COVID-19 actually tested for a sequence that can be found in the human chromosome 8. I came to the conclusion once I learnt about it that it was all complete nonsense. Also, neither Ike nor Kaufman believe in the existence of SARS-CoV-2 virus, nor in the reliability of the tests or the reality of any pandemic caused by the virus. They have enthusiastically jumped on the chromosome 8 claim. Chromosome 8 is one of the 23 pairs of chromosomes in humans. People normally have 2 copies of this chromosome. It spans about 140 million base pairs, the building material of DNA, and represents about 4.5 and 5% of the total DNA in cells. About 8% of the genes are involved in brain development and function, and about 16% involved in the production of cancer. A region of about 15 megabases that appear to have a high mutation rate. This region shows a significant divergence between humans and chimpanzees, suggesting that its high mutation rates have contributed to the evolution of the human brain. But Ike and Kaufman argued a while back that the PCR test might detect exomes instead of viruses, or that viruses and exomes are actually the same thing. But now they give another twist to the continuous story of virus denialism, the effect that both probe and primers have to work together to get a test result, and simply pointing to a similarity of one of the primers and a piece of the DNA, it just doesn't make any sense. It's kind of proving that he doesn't even understand it himself, and that they're both promoting stuff that they don't even understand themselves. Search the nucleotides of the HCOV 2019 human coronavirus for the forward string and you will not find it. Then research the reverse that I posted here and you will find it every time. So the fact that CT, CCC, TTT, GTT, GT, GTT, GT somehow matches a tiny part of the human genome, it does not matter. It is not the sequence the test looks for in the first place. The sequence the test looks for is whatever exists between the forward and the reverse primers. These sequences are very close together in the SARS-CoV-2 genome. David Icke and Andrew Kaufman lack even the most basic understanding of the PCR test and the whole genome sequencing in general. They are just selectively misreading whatever they think they understand from science to find confirmation of their own sort of narratives that they want to push forward, when in fact it's just nonsense. Kaufman also claims that viruses actually didn't exist. There is no evidence for a virus. Scientists had mistaken very small cell particles called exomes to be viruses. But exomes, far from being harmful to our health, are in fact helpful because they help us to get rid of toxins. He said if it existed at all, was a variety of ailments mostly caused by environmental factors or stress. Some viruses may look like exomes under the microscope in some phases of their life cycle, but that doesn't apply to all viruses everywhere. Viruses differ widely in their visual appearance, and more importantly, relying solely on a microscope is old school virology, since genomics and bioinformatics have now taken over. Everything under the sun, from animals to plants to fungi to viruses to bacteria, even the most primitive archaea, has been sequenced these days and aligned with the larger tree of life. The virus has never been isolated. As a consequence, the genome sequences of this particular coronavirus, which are now available, the majority of all this nonsense science, such as germ theory denialism, usually involves arguing Louis Pasteur's model of infectious disease, which was wrong, people say, and that Antoine Béchamp was right. In fact, its origins are rooted in Béchamp's empirically disproved in the context of disease theory of pleomorphism. Another obsolete variation is known as terrain theory and postulates that diseases' tissues attract germs rather than being caused by it, which stemmed from many other people later on in the 2000s, such as Stefan Lanka, the core argument of Lanca's seems always to be that viruses had never been isolated, and the electron microscope photos are in reality photos of normal cell particles. As for Stefan Lanka, in a court case, he was ordered to pay €100,000 to David Bardens, who proved the existence of measles in 6 different ways. Lanka then appealed, changing the goalpost that it didn't meet his criteria. The court case ordered that the measles had been proven to exist, but it didn't meet Lanka's criteria, and Lanka didn't have to pay the £100,000. He then proceeded to go around the internet and spread information which was embellished, regurgitated, misunderstood, and spread out for the last 5 to 10 years that no virus has ever been isolated. Ike claims, among many other things, that there is a connection between the COVID-19 symptoms and 5G, the fast internet platform soon to be rolled out in many countries and already operative in some others, including China. He states, with 5G, life as we know it will be over. COVID-19 doesn't exist, and the real disease is caused by so-called exomes, particles expelled from cells due to radiation damage. But as I have stated from January, February, and in many videos, that the 5G propaganda was to be avoided, and yet another subject people haven't fully researched and just took on all this nonsense as gospel truth, as they usually do. It was revealed by a number of people in late August that someone had tracked all the original 5G causes COVID conspiracy theories from a Twitter address tracked to an address in Brussels, where the EU's horizon is situated, all between March the 27th and April the 4th, with over 10,000 tweets, all from one channel, and then rolled out by paid agents like Mark Steele.
People seem to think that microwaves they associate with the microwave oven in the same way they associate radiation with nuclear radiation. They associate the word microwave with the microwave oven. The only thing that's connected there is the microwave oven uses 2.4 gigahertz. Let's put that as 2.4 gigahertz. It's round about there in the spectrum. And there's another device in your home that also uses 2.4 gigahertz. It's your Wi-Fi router. Oh, it's also looking round for a phone. It's also your phone that communicates the Wi-Fi router at 2.4 gigahertz and 5 gigahertz, and it's also your laptop that communicates at 2.4 gigahertz and 5 gigahertz with the router. Router in Britain, router in America. So it's worth mentioning 2.4 gigahertz, 5 gigahertz. 2.4 gigahertz is exactly the same frequency as your microwave operates at. One of those devices is designed for cooking roast potatoes, and the other is designed for communicating with your computer and transmits much lower power. One transmits thousands of watts, one transmits milliwatts. Keep in mind, this is visible light in here, and it's getting longer and longer and more harmless, more and more harmless. Oh, there's the mobile phones. So it's not even remotely. Then we're going down here to, well, for those of you who like such things, the low frequency resonant earth waves, which aren't exactly going to kill you because they're around us all the time. So I regard the electromagnetic spectrum below the visible section, I regard that all as completely safe.
Question 1. Where did the SARS-CoV-2 come from? Pretty obvious. It wasn't created in a lab and purposely spread. It's pretty obvious and has been debunked by a number of people that it doesn't exist, or it's 5G, or Chromosome 8, or false testing. It's none of those. But I'll only leave 2 questions: naturally derived and naturally spread, or naturally derived, and then later modified in a lab. I personally believe it was naturally derived, naturally spread, and they've built up a vaccine and pharmaceutical industry around this entire study for the last 25 to 30 years. They've had a number of companies, Chinese, Canadian, British, American, all studying and funding and paying for virology research in countries such as China. They've been, really studying hard the effects of what these things can do to humans and how they could manipulate the situation and, like I just said, create a massive corporate machine all around the study of this small thing. So this is kind of my chain of how it happened from start to finish. It was naturally derived from a distinct cave outside of Wuhan, China, around about 2013, and possibly even longer. People like DARPA, Michael Callahan, Shi Zhengli, they were all studying bats within caves and villages around these caves for 20, maybe 25 years. Plenty of evidence to suggest, and there's plenty of scientific reports from these throughout the years of their progress of what they've been doing. Poor lifestyle practices slows the process of converting angiotensin 1 to angiotensin 2 in the blood, which makes it a perfect host for the spiked proteins of the SARS-CoV-2 sequence to enter. They figured this out around about 15-20 years ago as well. So this was another part of the big study. Gates Foundation has put 13.5 billion to a number of companies, organisations and media outlets to build this massive web of sort of vaccine, pharmaceutical companies that are going to save the world at a huge cost. And as you've seen with the lockdowns and the trillions in bank loans, they've made a massive amount of money. And then the drug companies over the next sort of 20 years will make billions from many different type of diseases and curing these diseases. They created the Event 201, predicting everything that sort of came true. This happened in October of 2019, which was sponsored by the foundation again, the Gates Foundation. The official outbreak happened in China in January 2020. The WHO waited until March to declare a world pandemic. Zhong Nashan took over the sort of Chinese health authorities again, like he did under the SARS outbreak. Dr. Ferguson of the Imperial College London epidemiology models predicted 4% deaths in the UK when it was lower than 0.04%. The WHO then endorsed these models on a global pandemic, and all the countries, apart from a number, say I think it was about 4 or 5 countries, didn't follow what the WHO said, which was led by Dr. Tedros Adhanom, who had been accused many years ago of genocide within his own country, Ethiopia, and causing vicious outbreaks there as well with cholera. There's lots of evidence to back this up as well. Moving on, they're using Nobel Prize winner Karimalis's RNA PCR test, which amplify and find certain proteins linked to the SARS-CoV-2. The thing that I'm putting across is almost everyone on the planet has had this SARS-CoV-2 now, if not more than once. So the PCR tests are finding it in almost all of us because we've all had it, and the chances are that at least 80 to 90% of us don't even know we've had it because it's not that bad. The government have been hyping it up and pushing society towards vaccines, which leads to the last point. Billions in revenue for the campaign for vaccines, drugs, pharmaceutical research and contracts. This is happening right now.